Hello students. So we will just wait for 5 to 10 minutes. Let all the students join. Then we'll start the session. So, hello students. So, let me start the session. So, others can switch off the camera. Siddharth can switch off your video. So, let me share my screen first. So, is my screen is visible? Can anyone just?
Yes, sir, visible.
Okay, so let's start the session. So, the subject is bioengineering and interface with biology and medicine. So, this is a week one session, the problem solving session. So, basically, in this session, we'll just solve and discuss the last year assignment questions. So, the topic for the days are why biology for engineers, and which includes for two parts. Part one consider why biology for engineers, then life processes in cells and its properties, and clinicians' perspective. So, let me introduce myself. So, my name is Ankit Kumar Malik, a PMR fellow from IIT BHU. So, this is my second year of PhD in pharmaceutical engineering and technology. So we'll keep this session as a discussion session. You guys can also uh, stop me in between, and if you have any questions, if you have any doubt, you can unmute your mic and speak, or else you can just drop your questions in the chat box. So we'll start from this question one of the last year assignment. So the question one was the high speed bullet trains like Shinkansen in Japan used to make sonic booms as they emerged from tunnels, which was problematic for the locals. To remedy this, the Shinkansen was
Excuse me, sir.
Hanji, yes.
This question was done in yesterday's class.
Actually, na, we are two TAs for the same course. So we'll both cover the same session, the same questions. Those who have wanted to discuss in this session, they can attend. Those who already
Okay,
covered
okay.
those doubts are clear, they can do whatever they want.
All right, sir.
The same questions will be discussed in both the sessions. So my colleague Pooja has completed the same questions yesterday, I think.
Yes, sir.
So let me begin. So this innovation evolutions the world's fastest bullet train. Pick the correct statements. So the statements are the sonic issues was addressed by a method called bioinspired engineering. Then speed of light of kingfishers was mimicked. Third option is the biological features mimicked was the beak's design for its natural efficiency to slice through the water to catch a fish. Option D is option A and C are correct, and option E is A, B, C, and correct. So yesterday you have attended the session. So those who would like to answer this question and explain it are most welcome. Anyone? You have attended the session last time, na? Yesterday.
Yes, sir.
So those who are wanted to discuss this statement, please welcome.
Sir, actually, option E is correct.
Option E is correct.
Yes, sir. Because actually, bio-inspired engineering, actually, they are inspired by the biological phenomenon and biological behavior of animals,
Okay,
actually. And this address the engineering issue like the sonic booms that disturb people were reduced by looking at the beak of the kingfisher, which silently catches the fish in the water without making any sort of noise.
okay. So have you attended the last yesterday session?
Yes, sir.
So what she told? Option D is correct, right? Hello?
Yes, sir. Option D is correct. Just got confused. That option
Okay.
D.
Yeah. So the option D, as per option D, A and C are correct. The sonic issues was addressed by a method called bioinspired engineering, and the speed of flight of kingfishers was mimicked. So we'll just explain in a bit. So option D is the correct answer. Sec- So what is biometric designs? Biometric design refer to a man-made processes, devices, substances or systems which imitate nature to develop new devices that enhance human life. Through the help of biomimetic systems, scientists, researchers, engineers, and business people have increasingly turned towards the nature for design and inspiration. So, what Shinkansen bullet train was? So, this was the fastest train in the world, so which runs on a speed of 200 miles per hour, which is its maximum speed measured till date. So it is located in the west of Japan. It was introduced in the early 1960s, that is 1964, and it is considered the most reliable high-speed train in the world till. So how a Kingfisher inspired a bullet train? So in Japan, they have these very fast bullet trains. They were getting so fast that the typical bullet shape was causing a loud boom sounds when these trains would exit typical train tunnels. But one of the engineers on the team trying to solve the problem was a bird watcher. So that engineer has witnessed a kingfisher bird diving down through the air, going into the water and creating very little splash. So he thought, I wonder if I could apply this principle to the shape of the front of the bullet train. And so they did. Model the front of the train like the kingfisher's face. It has a pointy part to it just like the beak of the kingfisher. And sure enough, when they tried out that new model, it moved through the without creating the boom, and it saved them 10 to 15% more energy because it was more aerodynamic. So as you compare the beaks of the kingfisher and the train's front portion, so the body length and beak's length in 0.17 meter and 0.09 meter beak length. So based on this design, they have designed the bullet train Shinkansen front portion to save 10 to 15% more energy. So how they mimicked it? So they found that engineer found that the kingfisher's secret move is the long beak. So they redesigned the front of the train using the beak of kingfisher as a model. So why? So just because of this mimic portion, it has a quieter train. It uses less energy. It moves faster. It they balance the energy and more comfortable. So these are the graphs and the estimate designed by the engineers during the design of the Shinkansen bullet train in Japan. So we'll move to the question 2. So the question 2 is, what would be the possible advantages of engineers knowing biology choose the correct combinations? So the question 1 is, so the combination 1 is growth in biorobotics, which developing artificial skin sensing skin which can detect pressure changes upon touch inspired by animal movements. Second statement is development of bioweapons creating the deadliest of virus to deal with alien attacks. The statement third is development of biofilters, wastewater treatment, cleaning large water bodies. So they have given the combination four combination here. Either 1, 2 or 3 may serve as a potential advantage to mankind. Option B. Only 3 can be considered as an advantage to humankind. Then option C states that both statement 1 and 3 are the advantages to humankind. And the option D states that 2 and third statement are the real life advantages. Anyone would like to explain this? Anyone?
Sir, again, it's option C because, like creation of bioweapons, it has a destructive potential, not benefiting people. In fact, creating more wars.
Okay. Explain it more. Explain it a bit more.
Uh,
What
and
is biorobotics and biofilters?
So biofilters, they'll use the microbes and like microbes feeding layer is established so that the microbes can grow in colonies and they can mm, trap the dust particles.
Okay.
And whereas in case of the touch sensor, it just senses the um, like change in heat, pressure, so that like the person's like bodily this thing is known
Okay.
connection.
Okay. What's your name, man?
Sir Siddharth.
Okay, Siddharth. So we'll discuss it in a detail. So the option 1 and 3 are the advantages to the mankind, which is biorobotics and biofilters. So what is biorobotics? So the growth in biorobotics, particularly the development of artificial skin sensing. So it is an exciting advancement that draws inspiration from the way animal perceives and respond touch in their environments. This innovation involves creating a synthetic skin for robots that can detect pressure changes, similar to the way animals use their skin to sense and interact with their surroundings. All of you guys must have seen that movie Robot. So the same concept was used there for skin sensing. So the concept of artificial sensing skin is rooted in the desire to enhance the tactile capabilities of robots, allowing them to interact more effectively with the world and respond to various stimuli. And by mimicking the sensory functions of animals, researchers aim to create robots that can navigate complex environments, handle objects with more precision, and improve their overall dexterity. So, the development of artificial sensing skin involves integrating sensors into the robot's outer layer, which can detect pressure changes, vibrations, and other tactile informations. So, these sensors are designed to function similarly to the mechanoreceptors found in animal skins, which respond to mechanical stimuli such as touch and pressure. So, this is the concept of biomimetic multi-layer structures for the robotic skin. Basically, it is a mechanism that how the skins or artificial sensing skin are prepared or made, how it works. So, in image A, it is a comparison between the biomimetic multi-layer structures based on hydrogel elastomers and human skin. Is my marker is visible? That pointer
No, sir. Actually, it's on the same slide.
in okay in the same slide. On which slide it is?
So, question one slide.
Okay, it's not changing.
Yes, sir.
Okay, you should have told me, na man. Okay, wait, let me reshare my skin. Wait,
Yes, sir. Now it's visible.
okay, now it is visible. Okay, okay. So I think in slideshow it got stuck. We'll not go in a slideshow. We'll just keep in the normal. So this is the concept of biomimetic multi-layer structures for robotic skin. So here it is the mechanism of how it works, how artificial sensing skin works. So here there are 5 diagrams which explain the biomimetic multi-layer structures for robotic skin. So in diagram A, it's a comparison between the biomimetic multi-layer structures based on hydrogel elastomer hybrids and human skin. So both structure consist of an elastic and tough outermost layer and a thick and compliant cushion layer, which sensing elements were also evenly distributed in the multi-layer structures. And in the figure B, it's the multi-layer structures which allows the robotic skin to achieve an appropriate material property and dimensions for the protection functions. And in diagram C, Because of the hydrogen layer is complete and electrically conductive, it can be used as a pressure sensitive material. Also, the dynamic tactile stimuli can be measured by detecting vibrations generated on the surface and transmitted through the multilayer structures. And in the figure D, it is a scalable design based on distributed sensing element with a large and overlapping receptive fields. And in the figure E, it is a biomimetic robotic skin which can be repaired with an adhesive after damage to be restored its structure and function. Then moving forward, so the option 3 was, the statement 3 was the development of biofilters, which is wastewater treatment plant cleaning large water bodies. So this is the schematic diagram of the biofiltration system. So what is biofiltration system? A biofiltration system is designed and fabricated such that it consists of six main unit operations, with namely a wastewater storage tank, feed tank, pre-sedimentation tank, bioreactor, post-sedimentation tank, and the treated water storage tank. So here wall 1, wall, totally 9 walls are here. So the development of biofilters represents a significant advancement in environmental engineering. So particularly in the realm of wastewater treatment and cleaning large water bodies, biofilters are specialized systems that utilize living organisms such as bacteria and other microorganisms such as fungi and all to break down and remove the contaminants from the water. So we'll move back to the questions. So these are two statements, which is growth in biorobotics and the development of biofilters are the damn advantages to the mankind so far. So this development of bioweapons cannot be the advantages to the mankind because it's the deadliest of virus to deal with the alien attacks. So far, we do not have any statements to uh, make advantages to the mankind of bioweapons. So we'll move to the next question. So the question is, my slides are changing now. My screen is visible properly.
Yes, sir. It's
Yes, sir.
visible properly.
Okay. So the next question, which is third, to check the packaged milk quality of a particular brand, you put a drop of milk diluted with sterile water onto a glass slide and stain it. It is then placed under a microscope at 40x magnification. What could be the best possible protection for the microorganisms mounted on the slide based on its morphology? So this is the diagram, and here are some bacteria are there. So which kind of bacteria and what are the best possible protection of this bacteria or micro? I'm not saying this is a bacteria, it's a microorganism. It could be fungus, it could be anything. So this options are, it could be a budding yeast and gram negative bacteria, it could be a gram positive bacteria, most preferably bacillus, and option C is a mixture of gram negative diplococci and gram positive protobacteria. And the option D is a mixture of gram positive cocci and a gram negative bacteria. What is the best protection from this image? Anyone?
So option D, because gram positive cocci, it is stained with gram stain and it is showing purple color as a result of the staining,
Okay.
whereas like the bacilli bacteria are showing gram negative stain because of lack of peptidoglycan, because of no peptidoglycan layer which holds onto the gram stain.
Okay. Good. So the correct answer is the option D, a mixture of gram positive cocci and a gram negative bacteria. So we'll go into details and I'll explain everything. Why it's gram positive cocci and gram negative bacteria. So here are the different shapes of bacteria. So we have here cocci, monococcus, diplococcus, streptococcus, tetracoccus, staphylococcus, cercina, and in bacilli, which is a rod-shaped bacteria, in vibrio spirilla. Then we'll explain everything now. So in cocci, cocci are the bacteria which are unicellular, spherical, and elliptical in shape. Either they may remain as a single cell or may aggregate together for various configurations. So like monococcus are called as a micrococcus and represented by a single discrete round. And diplococcus are the cells which divides once in a particular plane, and after division the cells remain attached to each other. And streptococcus are the cells which divides repeatedly in one plane to form chain of cells. And tetracoccus are the cells which are of four round cells which divide in two planes at a right angle to one another. And moving to the rod-shaped bacilli. So bacilli are the rod-shaped or cylindrical bacteria which either remain single or in pairs. And vibros are the curved comma-shaped bacteria and represented by a single genus. Then the last spirals. Then spirally are the types of bacteria which are spiral or spring-like with multiple curvatures and terminal flagellas. So these are all about the structures of the bacteria. Then moving forward, the gram-positive and gram-negative things. So here is a schematical diagram of the mechanism, how it works, how gram-positive bacteria structures, how gram-negative bacteria structures are there. So the key distinction between the gram-positive and gram-negative bacteria lies in the composition of their cell walls, a fundamental characteristic identified through the gram staining technique. So gram-positive bacteria possess a thick peptidoglycan layer in their cell walls, which retains the crystal violet stains during the gram staining process, rendering them purple under a microscope. In contrast, the gram-negative bacteria have a thinner peptidoglycan layer sandwiched between the outer membrane and an inner membrane, preventing the crystal violet stain from retaining and causing them to appear pink. And gram-negative bacteria often exhibit greater resistance to antibiotics due to the presence of the outer membrane, which serves as an additional barrier to this. The differentiation between gram-positive and gram-negative bacteria aids in microbial classification and is crucial for determining appropriate treatment strategies. Then this is the staining process. So staining process covers in four steps, which starts from the primary staining of crystal violet, then followed by the mordant, which is grams iodine. Then third step is decolorizer, which by alcohol or either by acetone. Then step 4 is counter stain by safranin or carbol fusion. So these are the step-wise method for staining the bacteria. So after staining, it gives two colors, either the violet or purple or pink or red. So by these identification, we can identify whether it is a gram positive or a gram negative bacteria. So here the gram positive bacteria appears violet or purple. On the other hand side, the gram negative bacteria appears pink or red. So get back to the question also. So here, according to the wait, uh-huh. According to the classification which I have given here, so this is a mixture of gram positive cocci and gram negative bacteria according to its stain and its structures and shape. Then question 4. So the DNA sequence and structural comparisons characterize all living organisms into three primary domains, which is bacteria, archaea, and eukarya. Environmental studies shows that the archaea are abundant and varied in soil, seas, and fresh water. In addition to harsh conditions, choose the incorrect statement about archaea. So here there is one incorrect statement is there and three correct statements are there. You have to choose the incorrect one. So the statements are organelles and other internal membrane bound structures are essentially present in the archaea cells. Statement B is archaea possesses a single circular double-stranded DNA found in the nucleus, a cell region. The option, the statement C is the archaea reproduce through fission, in which each cell divides into rod and replicates its single chromosome. And the statement 4 is prokaryotes, particularly archaea, can endure harsh environments through the that are unfriendly to the most living beings. So anyone would like to answer this question? Then we'll discuss and how it works, which statement is incorrect.
Sir, option A, the first statement is incorrect.
Okay, And the reason?
So because archaea, um, the only difference in between archaea and bacteria is that they can like sustain in harsh environments,
Okay,
and uh, so from bacteria it evolved into archaea, and from archaea it evolved into eukarya.
okay,
So that's the that's why these bound structures are not present.
okay, okay, Siddharth. So I think my animation is not working. Just tell me if my screen is visible now or slides are changing or not. Slides are changing or it's on the
Um,
same slide.
so seems like.
Okay, after going to the slide show now, it's not changing the slide. Huh. So option A is the incorrect statement. So we'll know how it is incorrect and how all the statements are correct now. So moving further. So these are the three primary domains of living organisms, that is bacteria, archaea, eukarya. So these are the primitive living world was classified into two primary domains. Initially it was two primary domains. Then moving forward it divided into three primary domains. So earlier it was like eukarya and prokaryotes, that is bacteria and eukarya. Archaea came at last. Based on the microscopic characteristics such as the presence or absence of membrane bound nuclei and other cellular organelles. So, American microbiologist named Carl Richard and discovered the third domain of life, which is archaea, based on the distinctive 16S RNA, ribosomal RNA signature sequence, which had long been characterized as a prokaryotic organisms. So this became widely accepted. Am I audible now?
Yes, audible.
Okay, okay. So this became widely accepted as a third domain in 1990. So similarly, so nature of the cells hypothesis on the origin of all cells from a common ancestor termed as a LUCA. So LUCA is one term called last universal common ancestor, also known as the theoretical ancestor of bacteria. So this question can come in this assignment, like what is LUCA? So the LUCA is the last universal common ancestor. So this is the ancestor of bacteria basically. So the conclusion drawn out of the research conducted by the scientists that led to the differentiation of archaea from the bacteria are as listed below. So this archaea like halophiles, anticellular thermoprotus, pyrodictum, methanococcus are the last ancestor of bacteria. Then moving forward, so the difference between the prokaryotic and eukaryotic cells. So at the chemistry, we'll discuss at the chemistry level or a molecular level. So at the chemistry level, the primary distinction between the prokaryotic and eukaryotic cells lies in the organization and compartmentalization of genetic material. So in prokaryotic cells are found in bacteria and archaea, lack a true nucleus and other membrane-bound organelles. Instead of their genetic material, a singular circular DNA molecule is localized in the nucleate region within the cell. In contrast, eukaryotic cells, characteristics of plants, animal, fungi, or protists, possess a true nuclei enclosed by a nuclear membrane housing linear DNA organized into multiple chromosomes. Further, eukaryotic cells boast membrane-bound organelles such as the endoplasmic reticulum, Golgi apparatus, and mitochondria, each compartmentalized to carry out specific cellular functions. So the chemical composition of the cell membrane and the nature of cellular process also exhibit variances, underscoring the fundamental differences in the molecular architecture between prokaryotic and eukaryotic cells. So moving further, so this is the difference classification in archaea, bacteria, and eukarya based on the cell wall, cell membrane, metabolism, and genome and ribosomes. So the question 5 is, so if you work in a laboratory where you are instructed to culture an unknown strain after 48 hours of staking in a culture plate, you observe the bacteria has an extended thread-like morphology as shown in the picture below. So what is the organelle called? What kind of functional structural benefit does it impart to the organisms? So the option A is organelle flagella and its function is gliding movement. And the option B is organelle, which is pili, and function its cellular metabolism. The option C is originally is flagella and its function is rotatory movement. And the option D is organelle is pili and function is ATP production. So anyone would like to answer this question with a statement or any brief description on this?
Uh, sir,
Hanji? Hanji, yes, yes, yes.
sir,
Yeah,
sir,
please
I
go ahead.
think option C.
Option C. And what is the reason behind option C?
Sir, because flagella helps in the movement
Okay,
of the bacteria, yeah, uh, to assist the cell to movement. मतलब it helps the cell to swim from one location to another location,
okay,
and it has a rigid filament which emerge from the cell, and its motion is like rotation. It's
okay,
its its movement is similar to like a propeller in a boat,
okay,
so that's why.
okay. Anyone else would like to join? Okay. So, what is your name?
Sakshi.
Okay, so option C is the correct one. So, organelle is flagella and its function is rotatory movement. So, we'll go in a bit basic. So, here one bacteria is there with bacterial cell, its fimbriae, its pili, and cell surface is flagellum. So, there are different kinds of flagellar arrangements are there called articross, monoarticross, Lophotrichus, Amphitrichus, Petitrichus, and Amphilophototrichus. So based on this of different flagellar arrangements, we have to identify which kind of arrangement, which kind of pili, which kind of flagella it is. So as you said, it has a rotatory movement. So flagellum has a rotatory propeller-like motion and a cilia as a back and front beating movements from its basal body. So going in a bit detailed, so the flagellar movements of bacteria involves the rotation of whip-like appendages known as a flagella, so which are vital to their motility. Bacterial flagella are helical structures composed of their protein, flagellin anchored in the bacterial cell membrane and cell wall. So the mechanism of flagellar movements is powered by the flow of protons and other ions across the cell membrane, which generates a proton motive force that drives the rotation of the flagella, that is rotatory movement. So the bacteria can exhibit different types of flagellar arrangements such as polar flagellation, Petriacus, which I've told in the last slide. Huh? So this remarkable adaptation is crucial for various bacterial activities including locating nutrients, avoiding harmful substances, and facilitating the colonization of host tissues in the pathogenic species. So the next question is the endoplasmic reticulum. The endoplasmic reticulum is a substantial dynamic structure serving many roles in the cell. Choose point out the correct functional and structural information for the organelle. So the information first is structural basis. On the structural basis, divided into two: rough endoplasmic reticulum, which is with ribosomes, and smooth endoplasmic reticulum, which is without ribosomes. And its function is to synthesis, folding, modification, and transport of the proteins. And the statement information B is structural basis. Structural basis through rough endoplasmic reticulum only, and its function to assign post-translational modifications in proteins. And option C is structural basis, proteins and RNA embedded through rough endoplasmic reticulum, which functions is to provide transmembranal movements of proteins apart from synthesizing them. And the last option D, which is the structural basis, is RNA and protein, and it is in charge of converting encoded signals from messenger RNA molecules into the amino acid based proteins. So, anyone would like to explain it a bit with some hard statements?
So option A, I think.
Option A is correct.
Yeah.
So, the reason behind option A is what are the reasons?
Yes, so because endoplasmic reticulum is divided into two parts, rough endoplasmic reticulum and
Smooth
smooth endoplasmic
endoplasmic
reticulum.
reticulum. Yeah.
A rough endoplasmic reticulum helps in the protein synthesis, and ribosomes are the protein factory, which are closely associated with the rough endoplasmic reticulum. And smooth endoplasmic reticulum helps in secretion of hormones like progesterone, and it also helps the in transfer matlab, uh,
Mm-hmm,
Golgi
mm-hmm.
bodies are also associated with the endoplasmic reticulum, so they transfer the molecules
Okay.
or proteins or the whatever they
Mm-hmm,
secrete.
mm-hmm. Got
That's
you.
why, according to me, option E is correct.
Exactly. Option A is correct. So we'll discuss about this for the option A. So there's the endoplasmic reticulum. The endoplasmic reticulum is a complex and extensive membranous network within the eukaryotic cells, which consists of two distinct regions: the RER, which is the rough endoplasmic reticulum, and the SCR, which is smooth endoplasmic reticulum. The rough endoplasmic reticulum is characterized by the presence of ribosomes on its cytoplasmic surface, giving it a rough appearance under a microscope. So these ribosomes are engaged in a protein synthesis, and the rough endoplasmic reticulum plays a crucial role in the synthesis, folding, and modifications of protein that are either secreted from cells or distant design. Hello?
Yes, sir.
Okay, okay. Just let me know if I'm not visible, or like my screen is hang, or screen got lagged, or if I'm not audible. Just please let me know. Okay.
Okay, sir.
Okay. So the smooth endoplasmic reticulum lacks ribosomes and is involved in diverse metabolic processes such as lipid. So this is a rough endoplasmic reticulum as per the option A, which is responsible for the synthesis, folding, and modification of proteins. On the other hand, the smooth endoplasmic reticulum is low. Actually, my
Yes,
screen is
sir.
getting lagged here. Wait, I think internet connection is not good. Hanji. So here the protein processes and processing and secretion is given here. So which assembles the proteins from polypeptides entering through the endoplasmic reticulum. So the endoplasmic reticulum serves as a dynamic and multifunctional cellular organelle, facilitating the synthesis and processing of biomolecules essential for the various cellular functions. Its intricate structures enables it to play a central role in maintaining cellular homeostasis and responding to the dynamic needs of the cell. So moving to the next question, question 7. So choose the cell organelle that are majorly involved in the intercellular transport. So here some options are there. So like ribosomes, nucleus, and Golgi apparatus, which organelles are mainly responsible for the intercellular transport. So anyone would like to answer this question with some strong evidences? Anyone?
Sir, it's the endoplasmic reticulum, Golgi apparatus, and lysosomes.
Okay,
Sir, what happens is from endoplasmic reticulum, the proteins get transported to Golgi apparatus,
okay,
and from Golgi apparatus, then through clathrin coated vessels, they get transported through lysosomes.
okay,
So then what happens is lysosomes then they are also known as suicidal bags. So wherever there is protein deficiency, they just undergo autophagy and release the protein.
okay,
Along with that, for the transport of from endoplasmic reticulum and Golgi apparatus, there are two vessels.
okay.
One is COP1 and COP2.
Exactly,
COP So COP1 is from endoplasmic reticulum to Golgi apparatus.
exactly.
COP2 is from Golgi apparatus to endoplasmic reticulum.
Okay, okay, Siddharth. The option B is correct. which is endoplasmic reticulum, Golgi apparatus, and lysosomes. So now we'll just look into what intracellular protein transport is. So intracellular transport is a fundamental cellular process which is responsible for the movement of various molecules, organelles, and vesicles within the confines of a cell. So this intricate system ensures the proper distribution of essential components to different cellular compartments, which enables the cellular functions and maintaining homeostasis. So the two primary mechanisms govern intracellular transport, which is vesicular transport and a cytoskeletal transport. And vesicular transport involves the packaging of molecules or organelles into membrane-bound vesicles that transverse the cytoplasm and fuse with the specific target membranes, which facilitates the exchange of materials between organelles or with the cell membrane. So, so here the cytoskeletal transport relies to the dynamic cytoskeleton system consisting of its microtubule and actin filaments to serve as tracks for motor proteins. So these motor proteins such as kinesins and dyneins moves along the cytoskeletal tracks, transporting organelles or vesicles to their designated locations within the cells. So the coordination of these transport mechanisms is crucial for cellular activities, includes secretion, endocytosis, maintenance of organelle dynamics, ultimately contributing to the overall functionality of the cells. So moving forward to the next question, this is question 8. Mm, Question 8 states that the primary functions of the organelle flagellum is to facilitate mobility and chemotaxis. Name the element that is involved in creating the structure of bacterial flagella. That is porphyrin, flagellin, flagtubulin, bacterioactin. So this is the most basic question, the most easy question in this assignment. So we have discussed this question also in the last some slides. So anyone would like to answer?
So option B,
Why
flagellin.
option B?
So
Do you have
this
some?
is a globular protein.
Mm-hmm.
Hello.
You're audible.
So this is a globular protein which arranges itself in a hollow cylinder form,
Okay.
and it's a principal component of the bacteria
Okay.
flagella. That's why
Okay.
it's present in the largest amount, I think.
Exactly. The correct reason. Exactly. Good. So the option B is the correct answer. So the flagellin is the element which is involved in the creating the structure of bacterial flagella. So moving to the next question. The question 9 have some statements which have the correct regarding the origin of replication among prokaryotes and eukaryotes. So the statement 1 is prokaryotes have a single origin of replication, and option second is all eukaryotes have a single origin of replication, and statement 3 is all prokaryotes have multiple origin of replications. And statement 4 states that eukaryotes have multiple origin of replications. So anyone knows about this? The origin of replication of prokaryotes and eukaryotes? Anyone?
1 and 4.
1 and 4.
So I think option two is correct to both 1 and 4.
1 and 4. Any reason? Any strong evidence behind this?
So because in eukaryotes, for fast copying,
Mm-hmm. And
and
option. Mm-hmm. Go ahead.
in prokaryotes there is only one origin because uh, it is characterized by repeated sequences. I
Exactly.
think.
Got it, got it. You're correct. You're going well. So, what is the origin of replication? So, the origin of replication is a site where DNA replication initiates, which exhibits notable differences between prokaryotic and eukaryotic cells. So, moving to the next slide. So this is the DNA replication in prokaryotic DNA. In prokaryotes, such as bacteria, replication begins at a specific site on the circular DNA molecule, which is called as its origin of replication. So the DNA is unwound and the double helix is separated by the enzyme helicases. And the single-stranded binding protein stabilizes the single-stranded DNA and primase synthesizes the RNA primers to initiate DNA synthesis. And the DNA polymerase is 3, which adds nucleotides to the third end of the RNA primer to form a new strand of the DNA. While DNA polymerase is 1, remove the RNA primer and replaces it with the DNA. So the leading strand is synthesized continuously with the lagging strand, which is synthesized in a short or a there's one term called Okazaki. Have you ever heard that Okazaki fragment? Anyone? Hello, am I audible?
Yes, sir, audible.
Sir, thus,
There's one term called that Okazaki fragment. So, anyone would like to explain what Okazaki fragment is?
yes, so,
Please.
sir, I think this is the short sequence of DNA nucleotides
Mm-hmm.
which link together by DNA ligase to create a lagging strand during DNA replication.
Exactly. Hmm. Correct. So, uh, this is all for the prokaryotic DNA replication. So, moving ahead for the DNA replication in eukaryotic processes. So, here the replication is much more complex and occurs in a highly regulated manner. The DNA in eukaryotic cells is a linear and in packaged into chromatin, which must be unwound and replicated in a coordinated fashion. Replication begins at a multiple origins of replication throughout the genome, which are recognized by a complex of proteins called the origin recognition complex, called ORC. The helicases enzymes unwind the DNA at each origin and replication proceeds bidirectionally from each origin, forming replication bubbles. Here the DNA synthesis on the leading strands is continuous while the lagging strand is synthesized in a short Okazaki fragments. So the RNA primers are removed here and the fragments are joined by the DNA ligase. The result is two identical linear DNA molecules, each of which is packaged into a chromosome and passed on to the daughter cells during the divisions. So moving back to the question. So according to these statements, so option statement 1 and 4 are the correct. Prokaryotes have a single origin of applications and all eukaryotic have a single origin of applications. And moving further to the next and the last question, the human proton project, which is called as HPP, the Human Proteome Project, is a global initiative led by the Human Proteome Organization that intends to change our understanding of the human proteome. Which are the major missions of the project? Choose the correct statement from the following options. So the statement 1 states that it's a collaborative effort including numerous research labs throughout the world. And statement 2 states that it is a goal to systematically map the whole human proteome by utilizing existing and new approaches. The statement 3 states that this is a project which had one primary aim to cover the information about the human epigenome which was completed in April 2003. And the last statement aims to discover the missing proteome of the human body and creates the groundwork for the development of diagnostic, prognostic, therapeutic, and preventive medical applications. So, which statements are the correct statement? Anyone would like to answer this? Anyone?
So statements 1, 2, and 4, option B.
Statement 1, 2, and 4. And why?
So because actually the question is about human proteome project, and
Great.
whereas the third option specifically is talking about epigenome, which is like related to genome, not proteome.
Okay. Okay. And statement 4.
So statement 4, it's related to proteome, and proteomes have various therapeutic, diagnostic, and preventative measures which are being used in modern medicine.
Exactly. Correct. So we'll just keep it in light that what HPP is, what is Human Proteome Project. So basically, this HPP focuses on addressing key challenges in proteomics, such as improving techniques for protein identification, its quantification, and its validation. So by creating a comprehensive and publicly accessible knowledge base of the human proteome, it strives to advance our understanding of cellular processes like diseases mechanism and potential targets for therapeutic interventions. The projects also emphasize standardization of methodologies and data sharing to ensure reproducibility and facilitate collaboration among researchers worldwide, and ultimately contributing to advancements in personalized medicines and biomarker discovery. Hmm. So, based on this, option 1, 2, and 4 are the correct statements. So, this is all for today. Anyone would have anyone any doubt? So we can discuss, or else we can meet in the next session, which will be on the next Tuesday. Anyone?
So clear.
No, sir.
Okay, good, fine. So you can leave the meeting and we'll meet in the next Tuesday.
Yes, sir.
Thank you, sir.
Okay,
Thank you, sir.